Our new lecture is about the preservation and transport of the specimen, the remaining part of the previous topic. So, in today's lecture, we will learn about the transport media which is used for the preservation and transport of the specimen. What are the different types of anticoagulants which is used during the preservation and transportation of specimens? And the third and the most important part is the shipping of infectious substances, how to transport, how to carry a infectious substance from one location to another location, from one country to other country, from one area to another area. So these are the main headings of the today's topics. Coming to the today's topic, the transport media. Transport media, as the name indicates, it is used to maintain the integrity, the originality of the specimen if there is any issue in the processing or the processing is late due to some reason. So transport media is used for this purpose, to maintain the originality, to maintain the integrity of the specimen. The transport media, it contains several substances which is useful for the inhibition of the multiplication of microorganisms. These substances, they inhibit the binary fission and multiplication of bacteria, so the bacteria is not overgrown and they are present in their original state and the original numbers. The commonly used transport media, these are the Stewart's media or Amy's transport media. Along with these media, there are some other type of media in which charcoal is used. This charcoal absorbs the fatty acid substances in the swabs which is used for the culture purposes, because these fatty acid substances, they are harmful for survival of the certain agents, for example Neisseria gonorrhoeae and Bordetella pertussis. So these fatty acid substances, these are absorbed by the charcoal, and the Neisseria gonorrhoeae or Bordetella pertussis, they are being survived from the toxicity of these fatty acids. And if there is any issue in the processing of these organisms and these specimens, so they are maintained for certain time in the transport media. In some cases, the specimen is directly inoculated in the culture media at the spot of the time specimen collection, because this will give us the pathogen, and if the specimen is not processed and not inoculated at the spot of the collection, so there may be issue with the isolation of the organisms. So that's why certain specimen, sometimes there must be inoculation at the bedside of the sample. The blood samples, they are usually put into a broad culture media after the collection, because blood may be coagulates and the microorganisms present in the blood, they are then difficult to isolate and they may die in this coagulation process. So therefore, when the blood is collected, as soon as possible it is directly poured and put into a broth culture. Synovial and peritoneal fluids, they are also inoculated in the blood culture bottles at the bedside. These fluids also coagulates and clots, so therefore these are also processed at the bedside. And if there is any other procedure which is to be performed on these kind of specimen, then the additional specimen, they should be sent to the laboratory in a containers beside the blood culture bottles for gram stain or for other wet preparation. Specimens for Neisseria gonorrhoeae can be placed directly into a commercially available transport system which is called Jambeck system. This Jambeck system having a selective medium, and also they can generate a carbon dioxide because they contain tablets which produces carbon dioxide. So the specimen of Neisseria gonorrhoeae which requires a little higher amount of carbon dioxides, they are then transported with the Jambeck system. If there is a nasopharyngeal swab for suspicion of boreal pertussis, then these swabs are directly inoculated after the collection on a specific selective media for Bordetella pertussis, that is the bordered Yango agar. This agar is selective for Bordetella pertussis, especially from the nasopharyngeal swab. So these swabs are also not preserved and they are directly inoculated on the bordered Yango agar after collection. Along with these specimens, certain specimens which are collected from the eye, especially the corneal scraping, they are also inoculated on a suitable media for the isolation of microorganisms, because they cannot be preserved or they cannot be put into a transport media, or there is a little chances of the isolation of the microorganisms if these samples are transported in a medium. So these are inoculated on the media as soon as possible. The issue with the specimens which are collected at the bedside is that there are more chances of contamination at the bedside, because in the environment and other with the formats, and there are more chances of contamination of the media and as well as of the specimen. So the technologist has to ensure the contamination free inoculation of the sample on the blood agar or any specific agar. Sometimes technologists, the microbiology technologists or the lab technologists, they are asked to help in the collection of the sample by the doctor or the physician, and during this collection these technologists may also monitor the appropriate application of the specimen onto the media, because if sample is taken properly and it is not properly inoculated on the culture media, so we may lose our organism and our culture may produce a false negative, or we cannot get any growth from that area which do not have any microorganisms. So the proper application of the specimen is also important, and the quality assurance process, it must be ensured by the microbiology laboratory. These quality assurance include the quality of the culture media, the quality of the processing, and the environment, and the post analytical any errors which possibly affect the performance of the laboratory. So all these quality must be assured by the microbiology laboratory. The anticoagulants which are used in microbiology, these anticoagulants are used to prevent the clotting of the specimen, and these specimens include the blood, the bone marrow, the synovial fluid, or any other kind of fluid which having the clotting factors. These anticoagulants are important because certain organisms, when they are clotted in a material or when they are bound in a clotted material, so they are difficult to isolate. Therefore, the anticoagulants are very important to stop the coagulation of the sample. And the type and the concentration of these anticoagulants are very important because certain anticoagulants, they have the antimicrobial properties. Therefore, we must use those anticoagulants which do not have any antimicrobial properties. The most common anticoagulant which is used in microbiology laboratory is sodium polyanethol sulfonate. This anticoagulant is used in a concentration of up to 0.025% weight by volume percentage, because if we increase this concentration above this limit, so Neisseria gonorrhea and certain other anaerobes, they are inhibited by the higher concentration of this anticoagulant. So the maximum concentration should not exceed the limit of 0.025%. And the ratio of the specimen to the anticoagulant is also very important, that if there is a little quantity of the specimen, so the SBS should also be in a small quantity in relation to the specimen. And if there is more SPS, so it also inhibit the growth of the microorganisms, or it may kill certain microorganisms. So these microorganisms do not grow in the excess of the anticoagulant. Therefore, the ratio of the specimen to the ratio of SPS, the anticoagulant, it is very important. The different sizes of tubes, they must be available to accommodate the adult as well as the periodic blood sample, and also the bone marrow sample or synovial fluid. Another type of anticoagulant, which is heparin, it is also acceptable in the microbiology laboratory, and this heparin is usually used for the isolation or for the viral cultures or for the isolation of microbacterium species from the blood. The anticoagulants which is normally used in hematology, that is citrated or ethylene diamine tetraacetic acid, these are not preferred in the microbiology laboratory because they have certain antimicrobial effects, and they are not used in the microbiology laboratory for these specimens. Now, the third and the last point of today's topic is the shipping of infectious substances. The shipment of clinical specimens and culture of microorganisms is governed by a complex set of national and international guidelines. These guidelines are issued by the different departments in different countries. In US, these guidelines are issued by the Department of Transportation and the Department of US Postal Service. Along with these departments, if specimen is shipped or transported from one country to another country or by air, so there is another organization, International Civil Aviation Organization. So if the infectious substance is transferred by the air, so this ICAO, International Civil Aviation Organization, which set rules for the shipment of infectious substances. These infectious substances must be packaged in a specific and leak-proof container to avoid any kind of leaks, any kind of spills, or the spread of these microorganisms. And these packages must be labeled to caution, to inform the handlers about the hazardous content which is present inside the packages, and they must handle these packages carefully. The purpose of this packaging is to save everyone, the employees, the transportation industry, and as well as general public from the infectivity, from the hazardous of these infectious substances. And this should be properly packed by the laboratory staff. This is the responsibility of the laboratory staff to properly pack the infectious substances and properly label the infectious substances which is to be shipped from one country, from one district to another district, or from one country to another country. If you are transporting the infectious substances, so these infectious substances must meet the requirements of Directorate of Transport's hazardous material regulations, and these regulations are published in the Federal Register Title 49 Code of the Federal Regulation. If you are transporting it either by train or by water, by air, or by the highways, so you have to follow the rules and regulation from point 171 to 180. Before shipment, the specimens must be triple packaged. If you are carrying, if you are transporting, or if you are shipping the infectious substance from one area to another area, first of all, the material is placed into a primary container, and that container must be watertight to avoid any entry of water into the specimen, or to avoid the contamination of water of the specimen by the water. So after that, the absorbent material is placed around the primary container. Then this primary container along with the absorbent material is placed into a second container, and that second container is also watertight. The secondary package is sealed and placed into a strong outer container which is made from a strong fiber board, and this fiber board and all the containers, they must be labeled with the instructions, and specific instructions must be followed by the lab staff for labeling the container as a hazardous material. This is the triple packaging of the infectious substance. This is our specimen, the green substance which is present inside this first vial. This first container is the primary container. This is the cap, and this is the biohazard sign which is present on this first container, and this cap is watertight, so the water do not enters into the specimen. This specimen and the first container is then enclosed in an absorbent material. This blue color is the example of the absorbent material. And then both of this first primary container and the absorbent material is this placed into a second container. This secondary container is a strong container and is also a watertight, and this is properly labeled with the list of contents which is present inside this container, and also a biohazard sign should be present on this. And all these substances is then put into a third container. Containers must include the instructions of holding the specimen, of the transportation of the specimen, also a special instruction for infectious substances, the name, address of the shipper, and also the important documentation. So this is the packaging of infectious substances. If you are transporting, if you are shipping an infectious substance from one country to another country. These lectures were prepared from the textbook of Diagnostic Microbiology and Monica Cheese Pro. Thank you for watching the video. Hope you have learned. If you have any question, you can ask. Assalamu alaikum and Allah Hafiz.